Hey everyone, and welcome to this explainer. Let's jump right into one of biology's absolute masterpieces, polysaccharides. Today, we're going to uncover exactly how microscopic tweaks to a chemical structure give a molecule literal biological superpowers. I mean, we're talking about taking simple sugars and turning them into either a massive battery for energy storage or building material strong enough to hold up a towering redwood tree. It's fascinating stuff, so let's dive right in.
Here is our roadmap for today. We'll be moving through 1. The basics of polysaccharides, 2. Starch for plant energy storage, 3. Glycogen for animal energy storage, 4. Cellulose and chitin structure, and finally, 5. A study guide summary.
All right, Section 1. The basics of polysaccharides.
To really understand polysaccharides, we honestly just need to look at the name itself. Poly means many. So a polysaccharide is basically just a complex carbohydrate polymer made up of many, sometimes literally thousands, of individual monosaccharide sugar units. Think of it like a massive chemical chain. The individual links in that chain are the monosaccharides, and they're locked together by what we call glycosidic bonds. And out in nature, these giant molecules basically have two main jobs. They're either storing energy for later, or they're providing vital structural support to an organism. So how do these tiny monomer links actually snap together? Well, a single cyclic sugar has a hemiacetal group. For these sugars to polymerize into a chain, that hemiacetal needs to become an acetal. Here is exactly how that plays out. First, a hydroxyl group is protonated and it leaves. Then that carbon is attacked by an alcohol, specifically a hydroxyl group from a completely different monosaccharide nearby. The end result? The hemiacetal becomes an acetal and boom, you have successfully formed a glycosidic bond. You just repeat that process a few thousand times and you've got yourself a polysaccharide.
Moving on to Section 2, starch, which is plant energy storage. Before we look at the starch molecule itself, we really need to look at its fundamental building block. When comparing alpha glucose and beta glucose, they look incredibly similar, right? But there is one tiny, absolutely crucial difference right on carbon 1. In the alpha form, the OH, or hydroxyl group, is facing down. But in the beta form, that OH group is pointing up. That is literally it, down versus up. But as you'll see in a minute, that one subtle directional shift completely changes how the molecule behaves in the real world. For now, just remember that starch is built entirely from the alpha form, where that OH group faces down.
Now, within plants, starch actually comes in two distinct forms, amylose and amylopectin. Let's compare them real quick. Amylose forms a straight, unbranched chain. It does this by linking its alpha-glucose monomers using alpha-1,4 glycosidic bonds. That just means the bond connects carbon 1 of one sugar to carbon 4 of the next sugar in line. And because of the natural angle of these bonds, amylose actually coils up into this tight, helical spiral shape. Then, on the other hand, we have amylopectin. Amylopectin has those exact same straight chains, but it also has branches. How does it do that? Through a brand new type of connection, the alpha-1,6 glycosidic bond. Connecting carbon 1 to carbon 6 creates a branch point, allowing entirely new chains to branch off from the main structure.
So you might be wondering, why does a plant use starch to store its glucose? Why not just leave the raw glucose floating around in the cell? Well, starch is practically tailor-made for the job. First off, it's insoluble. That means it doesn't dissolve and mess up the delicate water balance or osmosis inside the plant's cells. Second, it's a massive molecule, so it won't just accidentally leak or diffuse out of the cell membrane. It stays right where the plant put it. And third, it's remarkably compact. Because those amylose chains coil up into tight little helices, a plant can pack a massive amount of glucose energy into a very small space. It's essentially the ultimate biological battery.
Section 3. Glycogen. Animal energy storage.
Glycogen is basically the animal equivalent of starch. Just like plant starch, it's made entirely of alpha-glucose monomers, and just like amylopectin, it uses both alpha-1,4 bonds for its straight chains and alpha-1,6 bonds to create its branches. But here is the critical difference between us and plants. Plant amylopectin only branches every 20 to 25 glucose units. Glycogen, however, is a highly branched structure. It has way more branches than plant starch.
In fact, glycogen branches every 10 to 12 glucose units. Why does this matter so much? Well, think about our lifestyles. Plants are stationary, right? They aren't running away from predators or actively chasing down prey. But animals are incredibly active. We have a much higher metabolic demand. Because glycogen branches so frequently, every 10 to 12 units, it creates vastly more ends or tips on the outside of the molecule.
So, when you're doing something strenuous and you need quick energy, enzymes in your cells can attack all those multiple ends simultaneously. They rapidly hydrolyze the bonds, snapping off individual glucose molecules for instant cellular respiration. It is a perfect example of a form-following function.
All right, Section 4, Cellulose and Chitin Structure. We're officially leaving energy storage behind and shifting our focus to pure rigidity and strength.
I want you to consider a real puzzle for a second. Why is cellulose, which is the stuff that makes up tough plant stems and towering tree trunks, so incredibly strong when starch, like a mushy potato, is so easily broken down? They are both literally just long chains of glucose. So how can the exact same building block create such vastly different materials?
The answer lies completely in that stereochemistry we talked about earlier. Remember? Unlike starch, cellulose is made from beta-glucose, where the OH group on carbon 1 faces up. But this creates a tricky geometrical problem. If you try to link two beta-glucose molecules right side up, their hydroxyl groups on carbon 1 and carbon 4 are physically too far apart to react with each other. So, what is nature's brilliant workaround? The beta-glucose flip. Every second beta-glucose monomer is literally flipped upside down. By inverting every other unit, the hydroxyl groups align perfectly, allowing them to finally react and form a beta-1,4 glycosidic bond.
And that single molecular flip is the entire secret to cellulose's massive strength. Because every other monomer is inverted, the resulting cellulose chain doesn't coil up like amylose does. It forms a perfectly straight, unbranched line. This straightness allows multiple long chains of cellulose to lie completely flat next to each other, and when they do that, weak electrical attractions called hydrogen bonds form crosslinks between those parallel chains. Now, while one single hydrogen bond is super weak on its own, thousands of them grouped together act like microscopic superglue, binding the chains into incredibly strong bundles called microfibrils. Those microfibrils then bundle up again into even thicker macrofibrils. The collective tensile strength of this entire structure is exactly what gives plant cell walls their rigid, bulletproof armor.
And hey, plants aren't the only ones utilizing this structural trick. Take chitin, for example. It's basically the insect alternative. Chitin is structurally very similar to cellulose. It also forms long, unbranched chains that provide immense structural support. But instead of stiffening a blade of grass, chitin is used to build the tough, crunchy exoskeletons of insects and crustaceans. So it's a completely different kingdom of life, but the exact same brilliant molecular engineering.
Finally, Section 5, the Polysaccharide Study Guide Summary.
Bringing it all together in this summary matrix, you can clearly see how everything connects. We have amylose and amylopectin making up plant starch from alpha-glucose, differing only when it comes to that 1,6 bond branching. You can see glycogen, the animal equivalent, taking that exact same branching to the absolute extreme for fast, accessible energy. And then finally, cellulose, standing completely apart by using beta-glucose, flipping its monomers, and forming those rigid, straight chains. I want to leave you with one final thought to really ponder. One of these molecules provides the literal calories we need to survive, while the other passes right through us as dietary fiber because our enzymes simply can't break it down. And the only difference between them is just a single hydroxyl group pointing up instead of down, forcing a chemical flip. Isn't it absolutely astounding how just one inverted chemical bond can completely dictate the physical reality of life on Earth? Keep that in mind next time you eat a potato or lean against a tree. Thanks so much for joining me on this explainer. Keep asking questions, and I'll catch you on the next one.